So we work on very tiny proteins called motor proteins. These are of the nanoscale. They allow things to be taken from one place to the other inside living cells. And we try to understand how these motor proteins and the transport that they bring about is important to degrade pathogens, bacteria, virus, parasites inside immune cells of your body, and also how these motors are important to secrete fat, to release fat from the liver into your blood. We were interested in this particular one. It's called the dynein motor. We found a few years ago that it is able to work in large teams. If you take a certain number of these dynein motors, their function is proportional to their number. This is not necessarily true for other motor proteins. What we were then interested in understanding, okay, if nature has made this motor so that it can work in large teams, how do you assemble such teams?
Hello everyone. I have some great news for you. Over last 90 days, the number of viewers have definitely increased in my channel. But most of you have not yet subscribed. Please do subscribe, and one subscription will immensely motivate me and my team to get back to you with some interesting and even better podcasts. So, if you feel that any of our podcast has brought some value to your life, please hit that button. This series of podcasts are mainly for the scientific enthusiasts, and our constant effort is to bring to you a knowledge which adds value, a knowledge which has quality, and a knowledge which empowers you. With that thought process, me and my team, we have been constantly working to get best information available within the scientific fraternity and bring it or make it available to you. Our bodies are intricate machines which work in combination with millions and millions of cells, and they keep us moving. From the majestic complexity of our brain to the intricate network of blood vessels and down to tiny molecular machines that power our cells, the human body is a marvel of engineering. As we go about in our day-to-day lives, it's easy to take for granted the incredible processes that occurs within us. We rarely stop to think about these millions and millions of neurons firing in our brain, or trillions and trillions of minute motor proteins which happen to work relentlessly in our body to make it functional. Motor proteins play a very crucial role in our day-to-day processes, including lipid regulation. And it can lead to several diseases like fatty liver or atherosclerosis, a condition where a part of your artery is blocked, which is so dangerous that it can lead to heart attacks. Motor proteins work in combination, as you can think them like very minute motors which are working in your body relentlessly to make it function efficiently. In today's episode, we are excited to have with us Professor Roop Mallik, a renowned expert in the field of motor proteins. Professor Mallik's research has significantly advanced our understanding of motor proteins and their impact on lipid regulation. Today, we will discuss and understand from him and get the latest insights about these motor proteins and explore the potential implications for human health. We will also deep dive into the life of a researcher. So, please join us in welcoming Professor Roop Mallik. This podcast will be a little bit different from my previous podcasts because here it will be primarily focusing on to your area of work.
Okay.
What you are actually working right
Okay.
now. It's because it is something very different and interesting area which not many few people are aware of.
Yes.
So primarily I'll try to focus on to that area aspect of it and then move on to a little bit wider research spectrum.
All right. Sure.
Right?
Yeah.
Sir,
Oh.
so to start with, because I saw most of your interviews, you say you are working on hyperlipidemia. Now, what is hyperlipidemia, sir?
Hyperlipidemia is too much fat floating around in your blood, circulating in your body, too many lipids, okay, hyperlipidemia. And the danger with that is that these lipids can be absorbed by different kinds of cells in your body. The most obvious one is your heart cells, cardiac cells, where once they get absorbed, they go and sit inside your arteries and can block your arteries and cause cardiovascular problems. That's not the only problem that it causes. In general, obesity and diabetes, these are, the invisible epidemics of today, perhaps much more serious than things like cancer or the different viruses that we talk about. So, uh, lifestyle changes are required, of course, but unfortunately, it doesn't work all the time. Some individuals are highly susceptible genetically to these kinds of high lipids in their blood, high amounts of lipids in their blood, and they might need other kinds of therapies. So uh, our purpose is to look at possible cures for abnormally high levels of lipids in your circulation. And to do that, we come from an angle where we really ask very basic questions at the level of cells and molecules. And if we understand those questions and find answers to them, hopefully we can at some point try to help people with such. So we have some leads on that, but these kind of attempts take a long time and they are quite difficult in my view.
Yeah.
And
Sir, as I understand, we always talk about lipids or more fat deposition in our body. So people always have first name coming in their mind is cholesterol. Is cholesterol always evil, or it has some positive side as well?
no, cholesterol is certainly not always evil. Cholesterol is required for giving the membranes in your body. There are all kinds of membranes. Cells are covered by membranes. Things inside cells are covered by membranes. All of these have a fairly high percentage of cholesterol, and that amount of cholesterol is required to make these membranes the way they are, to organize different kinds of lipids, proteins on these membranes. And for most biological functions, you require cholesterol. I mean, there's a reason why our bodies make it. But too much of everything is bad. So, yeah.
Yeah. So in that case, you're saying unit is more than the optimum level, then only the problem, or you can say the repercussions can be.
Repercussion. Yeah. So, like with everything else, more than an optimal level is bad.
True.
Yeah.
Agreed.
Yeah.
Now, in cholesterol also, so there are also segments, LDL, HDL,
Yeah.
VDL.
Yeah.
So all the categories, what we understand is LDL is something which cannot be broken down. So
So these are not different types of cholesterol, LDL, VLDL. These are different kinds of, small little bags inside your body you can think of, where there are different amounts of cholesterol of different types, not just cholesterol, other lipids as well.
Yeah.
Okay. So your, one kind is very low density lipoprotein. It is very low density because there's lots of fat in it. That's why it's low density. And gradually, as these very low density lipoproteins pass through different parts of our body, the lipids get absorbed, different lipids to different extent in different places. And when they get absorbed, the density changes. And that's why you now have a high density, low density, etc., etc. Yeah.
Sir, as lipidemia we were talking of, now atherosclerosis is one, right? You have problems, and like the cardiac arrests and all that, those things also we see.
Yeah.
Triglycerides, increased level of uric acid, those are all side effects of hyperlipidemia. But what I understand initially is like one is a genetic makeup of an individual which contributes to more
Right.
lipid deposition.
Right.
Other can be also a dietary contribution or environmental factors.
Yes, of course. Yes. So people who have genetic disposition towards these kinds of diseases are, they suffer at an earlier age very often. Okay. But generally, dietary people who don't have a genetic disposition, but they do not have the right eating habits or they don't exercise or these kinds of things, they may have problems sometimes at a later age. I mean, it's hard to define something very precisely here. It depends on the individual as well.
So in Indian population, sir, obesity can be labeled to some extent because of the genetic makeup and the food habit cumulatively.
Yes, both are responsible. Like, not just in the Indian population, everywhere else. In some families, you will see that there is a propensity to obesity that can be genetic. I'm not an expert on that.
Okay.
So my expertise is so. I mean, I'm not an expert on hyperlipidemia and the clinical aspects of that. We try to find the mechanisms in the molecules which eventually may lead to such conditions.
Correct.
Yeah.
Correct. So as you've given already the lead for this,
Yes.
so then coming to the motor proteins,
Okay.
which is your
Yeah.
actual
Right.
area
Right.
of
Right.
expertise.
Right. Right.
So if you can say, what are these motor proteins? You say, if mentioned, is there like small machines working in our body?
Yeah. So there, broadly, you can think of these proteins which there are several categories. One is where I think most of these motor proteins are able to generate force. Okay. And that if you are able to generate force, then you can do work. That we all understand. And some of them generate force, they sit at one place and they go round and round. Okay. So that's like an electrical motor. Okay. So they can rotate, and that rotation is converted into useful work, just like in your scooter or in your car. Okay. You use rotation of the engine to turn your wheels. Just like that, rotation of such a protein in your body is used to make different kinds of molecules. One obvious example is adenosine triphosphate, ATP, which you need to make energy. So you make these little packets of energy by rotating a molecule somewhere, and you bring the different components together and you make ATP. That ATP is then circulated around everywhere, and you use it for your heart to beat, for example.
Correct.
Okay. So that is one kind of motor which is rotational. Okay. There are other kinds of motors which don't rotate but which move in a more or less a straight line. Okay.
Okay.
So they are like when you are walking around. So that is another kind of motor. So our interest is in these latter kind of motors, which are called translational motors because they go from one place to the other. Okay. So uh, now why are they going from one place to the other? Because when they are doing so, these translational motors, they are required because they can generate force. Your body can harness that force to do many different kinds of things. One very obvious one is to divide cells. Okay. When cells divide, they are dividing all the time in your body. When cells divide, you need to separate the contents of the cell, of the nucleus, into roughly two equal parts, sometimes not equal parts. And that's another interesting question. And then you can make two out of one. Okay. That is the basic reproduction in some way. So there you need these motors to segregate different components. One obvious one is DNA. Okay, so you have to segregate the DNA. The DNA has to duplicate, and then you have to give equal DNA to both the cells so that they can carry on and keep repeating the same thing. So that's one place where these kind of translational motors are used. Another place is if you look inside cells, there's amazing dynamics inside cells. Things are fluctuating, things are moving around. Bacteria which get inside your cells, virus, they all need to move around inside cells. They can't just be sitting somewhere because biology is about things interacting with other things. Okay, just like our social life as well. Uh, and if things don't interact, they can't be interacting with other things if they are just sitting around somewhere. So you need to move around to meet somebody else. You need to exchange things. So when that happens in biology, subsequent to that, you have chemistry. Okay. So, but before that, you need physics. So the physics part is how you move around to one place to the other. And then beyond that is the chemistry, and the sum total of all this is biology.
Biology.
Yeah.
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 And that is where I see, most of the people, like even you also, you had a physics background and then you moved on the biology to apply all your physics knowledge.
Yes.
Right.
Okay.
That's how it
Yeah.
works.
Yeah.
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 Sir, as motor proteins, like, okay, you said you are interested on the translational movement, but how do you study these motor proteins?
Yeah. So there are many different ways of doing it. Uh, one would the easiest way to visualize it is that okay, these things move from here to there, so you can observe that motion.
You can try to stop that motion from happening and see how much force you require to stop it. That's how you can understand how much force is being generated for that motion, right? So, but there are many other less obvious ways where you try to purify these proteins, you try to understand, so nothing works in isolation. So these motor proteins must again interact with several other proteins, lipids, to do their job. So you need to do a lot of, molecular biology, biochemistry, proteins, lipids, all of these. This is wet lab work, which you do on the bench. So that part also we have to do. Sometimes we have to purify these proteins from animal tissue, not sometimes, very often. So because our interest is really understand what this molecule working at the nanometer level does to the animal. Okay, so then you have to get it from an animal source, and the animal can be in a certain condition or in a different condition. Between these two conditions, how is the motor protein working different? That you can't just address by observing motion. You need to see what are the other.
What are these conditions precisely?
The most obvious one could be whether you've eaten or not eaten, for example. Okay, so between those are two different metabolic states. Okay, so that is one of the things that we have done. Maybe somebody who, suppose you look at people, who have obesity or don't, who are not, people who are susceptible to that, disease versus healthy condition. This can be also,
Okay.
this is another possibility. Yeah. Okay, if you administer a particular drug, okay, and you don't administer a drug, then what is the difference? These are all different possibilities.
So then classification of this type of motors, one is because the translation and because the movement.
Yeah,
One you said as a rotation.
correct.
And any other types?
No, many other, even within these movement ones, translational ones, there are many different types. Even within the rotating ones, there are many different types based on where they are present inside cells and what is their purpose. So, for example, the ones which are translating like this, they so they need some rigid object on which against which they generate reaction. You can't walk on a slippery floor, right? So they're just like that. They need to generate traction against something else, and so they usually walk along tracks. Okay, and these tracks are actually polymers inside the cells of your body, and these polymers can be of different kinds.
Okay.
So there are polymers called microtubules, there are polymers called actin. So there are motors which generate force against actin, there are motors which generate force along microtubule. Now, even along the motors which generate force along microtubule, there are hundreds of different types. Okay, and we know very well about maybe 5 or 10 of those, but there are so many different varieties, and many of them we just don't know.
Yeah. So do they work in combination with each other or in isolation? Like
No, they both
both work.
both
Huh?
possibilities are there. They sometimes they work in combination. Large groups can work together.
Yeah.
Sometimes they work in depending on the geometry of the that
Right.
is, yeah,
Right.
yeah,
But sir, again, like how are, say, one is, as you said, this there is one particular I was reading about dining.
yeah.
That is one particular very interesting type of, out of all this motors,
Okay.
and they have a particular job. Now, one place I was also going through that their numbers
Yeah.
actually define their, like, how much work they are going to do.
Right, right.
So
Right.
that is only true for dining. It is not true for the other motors.
It's not so black and white, really. Okay, so there can be variations, and again, honestly, very detailed studies have not been done on all kinds of motors. What we do know well is that these dynein motors can work well in large numbers. Okay, just saying that that's the only motor which can work well in large numbers would not be right, because we just have not studied. But there is evidence that there are some other motors which are not able to work very well together. Okay, if you put two, they don't do much better than one.
Okay.
Okay, but if you take the dinings, if you put 3, 4, 5, 6, up to 10, we have checked, they can do much better compared to one.
By two.
Yeah,
Okay. So that's the interesting part,
yeah,
because we have been focusing on to one. But again, my question is, sir, why I'm just trying to understand in a very simple language.
Yeah,
way.
yeah.
Because these are, if you can imagine, as like tiny robots.
Correct.
But the robots are individually doing their small bit of work
Yes.
to accomplish a bigger task.
Yes.
But anyone is controlling the overall class of these motors or group of these motors.
It's about time scales. Okay. Yes, of course, somebody has to control what these. They can't just let them do whatever they want. But that control, I think, happens on over longer time scales. Okay, so let me give you an idea. Like, so these, if we talk about robots, these which move something from here to there, they are taking steps.
Yeah.
Okay, so they are walking just like we walk, and the typical time taken for a step can be as fast a few, let's say 10-20 milliseconds. Okay, so it can take about 100 steps in a second.
Oh wow.
Okay, so that's really, really fast. And I don't think you can, there's much control over a time scale of milliseconds. They just go about walking. But over longer time scales, if something else comes and attaches to this motor, or it enters a different area inside the cell, it can start to work differently. So like I told you, like whether you've eaten or not eaten, right? Fed versus not eaten. Those things happen over longer time scales. There you can control something else which will come and attach to this motor, or it finds itself in a different situation. There it will start working differently. So yeah, so the control, I believe, happens over a very different kinds of time scales. Okay,
And it varies from person to person also, maybe.
I don't know the answer to that. It may, if depending on healthy versus not healthy or different things, but we really don't know the answer.
Now, coming to this a little bit more on the therapeutic angle to it.
Yeah.
So these robots, tiny robots, what you are saying as motors, motor proteins can be applied as a therapeutic to somebody who is already suffering from certain.
You could, so these are all still science fiction, but you could think of some possibilities where you package a drug onto a motor protein,
Yeah.
and if you can program it to go to a desired location, that is one possibility. But perhaps what can be more, like there is a motor protein called kinesin. Okay, there are many forms of kinesin again, and mutations, changes in these proteins are known to be relevant. They're known to be correlated with certain kinds of cancer. Okay, so if you are able to block some of these kinesins from doing, so for example, there are certain kinesins which are required for division of cells. Okay, if that kinesin goes into overdrive, okay, then cells will start dividing more. What is that? Cancer.
Yeah.
Right. So if you have a tumor, your cells are dividing more, so you have a tumor. So if you can, if somebody has, let's say, a genetic disposition or some other reason because of which the cells are dividing more and you have a tumor, one possibility is to figure out what kind of motor is required for dividing these cells more and more, and try to block it.
Yeah.
These are possibilities. Yeah. So we are trying to address this in some other way, where we try to look at what we started with hyperlipidemia. Okay, too much lipids in your blood. Can we control the function of actually a kinesin motor to perhaps reduce the amount of lipids in your blood? So yeah, so it is possible, but still, yeah, one needs work. Yeah. Yeah. It's not far-fetched, but people are doing things. Yeah. We are not too far. Yeah.
Biomarkers are there for this,
For
for the motor proteins?
biomarkers,
Or is there anybody working on this, like to identify, like how do I understand what all motor proteins are working in my body?
oh, there are hundreds and hundreds. So there is. So you are trying. You are asking me if one kind of motor protein is working more in, let's say, a diseased person.
True.
Then is there? How do you know it by a biomarker?
Correct.
I don't know of anything like that. Right. I don't know, because usually these pathways are very complicated. Something happens, then 10 different things happen. So it's not easy to make inferences. Yeah. So therefore, to try and address that, what we try to do is try to do experiments at many different scales. Okay, we try to do it in animals. Whatever we are thinking, we try to make it, on the tabletop.
Yeah.
And try to see if it still repeats. So then try to make a minimal system in which you can repeat something.
Correct.
Okay, which is happening, let's say, in an animal. Most often we are not successful, but sometimes we succeed.
Yeah.
Then we are more sure about the thing.
Yeah. So the other aspect was if you have any pathogens, that also motor protein helps regulate.
Yeah.
So can we just explain that mechanism?
No, let's say, pathogens, let's say, viruses, when they enter your lung cells, okay, when you inhale something, we went through all that recently, but so those viruses are taken in by cells lining your, lung epithelium.
Yeah.
And those by a process called endocytosis. Okay, so the viruses get in, and the viruses have a coat on themselves, or called a capsid. Okay, and then the viruses have proteins on that capsid which try to attach. We have affinity for motor proteins.
Correct.
And so that's how the virus finds its motor.
Okay.
And once it finds its motor, that typically what happens for many viruses, they come with their own DNA and RNA, but they don't have the ability to replicate it. Okay, so they need to send, once it gets inside a cell, it needs to send its DNA or RNA to the nucleus, where the host cell, your cell, the machinery of your cell will replicate
Correct.
the DNA of the virus. And so you will have more viral DNA.
Right.
So to do that, the virus which is coming in from outside needs to get to the nucleus. So for that, it uses a particular kind of motor which will take it from the periphery of the cell to the nucleus. Once it has replicated inside the nucleus, it needs to now escape from that cell to infect more cells. So then what happens is that all the virus particles that have been made by the initial process, they now get onto the other kind of motor, which takes them to the periphery of the cell. And then they can, lyse the cell or get out by other methods, and then they can repeat the whole process again. So they can manipulate
They are actually manipulating and using our machinery.
Yes, so they have, by evolution, by random evolution over the years, they, whatever has worked for them, they persist with that, right? And that's how they figured out. So the ones which couldn't figure it out died off.
Yeah.
Yeah.
So like we have seen motor proteins are in the market for work has been going on for last 10 years roughly now.
No, much more than that.
Much more than that.
Yeah,
So what do you see like evolution has happened or development happened in this research area, and what is the future prospect or areas of work which are still pending or which can work? Some we'd already discussed about maybe the therapeutic part.
So they, now we have much more improved technology. I mean, you are in this field, you know much more than me, to actually image these things, even sometimes in deep tissue, in small organisms. So I think what now needs to be done is to find out all this knowledge about the molecules and the physics aspect of this that we have accumulated over so many years in tabletop assays where we have purified systems, how to apply all this to cells and animals.
Correct.
Okay, that and try to understand it not at at a more deeper level. So that will probably is the next.
True.
Yeah.
True. Sir, coming in this line only, like what can you just explain optical tweezers or optical trapping technique?
So the optical trapping technique uses a very highly focused beam of laser light, uh, and in that light beam you can actually hold small particles. Let's say a small bacteria or a red blood cell or a small plastic bead. And why is that useful? Because it is a non-contact way of generating force, right? Because otherwise, if I want to generate force against you, I will push you. Okay. But if I shine enough light on you from far away, I can push you away.
Yeah.
That is a possibility. And that is because the light contains photons which have a certain amount of momentum.
Yeah.
So I can use that momentum to generate force at a distance. Okay. So So by now, of course, I mean, to generate enough light to push you away is going to be very difficult. But if you are a small bacteria, then I can generate enough light with available laser sources. So in an optical tweezer, you generate such a focused beam of light typically using a laser and small micron size particles, let's say a bacteria inside the cell or other kinds of endosomes, phagosomes, lipid particles. These you can exert force on these particles. And of course, that has many possibilities once you can do that. And our purpose to do that is these kind of particles, endosomes, bacteria, they are being carried around by these motor proteins. And if I can exert force with a light inside a cell or at a distance, then I can stop the motor from moving and measure, therefore, how much force is required to stop the motor. If I can do that, then I can measure how strong the motor is.
Yeah.
So that's the
That's an indirect way of finding it.
Yes, indirect way of finding out how much forces this motor apply, how much, I mean, the force is a measure of its activity.
Yeah.
So that's how we measure.
But at times, two motors, they can work in different directions.
They can work in different directions, yes. So then you have more complicated data from these optical tweezers. So those also we try to generate and analyze that kind of data.
Yeah, because the force is there, but they're canceling each other.
They are cancelling each other, or the opposite forces may not be generated at the same time. There may be some kind of a temporal coordination when this one goes versus that one goes. That is also an interesting aspect.
Okay, okay. So so apart from this optical tweezer method or this optical trapping method,
Yes.
any other technique you use it to study these motor proteins?
Yes, we use direct imaging visualization. We also have in the past tried to use magnetic forces to do this. And we do a lot of, as I said, wet bench work where you can make the system in a different manner and then take it to these biophysical studies. You can prepare the system in a different manner, give it certain other proteins which this motor can interact with or not interact with, uh, extract these motors from uh, different healthy or a diseased tissue, for example, or metabolically different states, fed versus fasted, and then you see how the motor works differently. These are things that we can.
Wow, that's interesting. So fascinating to listen to. But I must say that it's not that easy as the way totally the way you have explained things.
Okay.
It's definitely not that easy, which I ask with my little knowledge.
So we all try our best. Yeah.
True, true. But what role microscope plays in this research area?
So you can make many kinds of adaptations to microscope. One simple example is that the optical tweezer typically is built on a microscope. So like we have a microscope here which we had to modify quite a lot. I learned those things when I did my postdoc earlier, and some of it I replicated here and some new things we made. And so you take a microscope and you open it up and you insert different components inside it. You insert another laser, and that's how you can build an optical tweezer around a microscope. Microscopes are also of many different varieties, like you're using a light sheet microscope with some super resolution abilities. So all of these, there's no answer for one answer for everything. Yeah.
So coming to us a little different aspect to it, your interest in this area, how you found it out?
I uh, I I joined for postdoctoral work with uh, my supervisor, who was also coming from sort of a physics background, and so there was a little. It's easy to converse, and he was working on certain other aspects of these water proteins. So that's how I got into it. It was nothing very planned or anything. That when I was 5 years old, I started thinking about water proteins. No.
True.
Yeah.
True. Usually we feel, no, as parents in this field, or I've been seeing other people, young researchers. I ask this question, like, what keeps you motivated every day? Because in this life of a research scholar, I understand, you have
No, it's not necessary that you will be motivated every day. Sometimes you are demotivated, sometimes, but you have to pick yourself up. Every day is not a.
what is that thing which keeps you ticking?
I see, if you have succeeded in the past, you have confidence in your abilities. Okay, you know how to maneuver around problems, and that's you have to believe in yourself.
Yeah, that's I
Yeah.
think is also a wise word. I'll also take it from you. Sir, then anything which for the research scholars you would like to say that so they also you have passed their life. So what is their motivation? What should be their keywords from you?
Their keywords should be, work hard, smart, and read. Okay, you need to know what others have done, not just do your own thing. Because then you are in for, when you start compiling your work, you will be in trouble if you have not read what others have done.
Yeah,
Yeah. So,
yeah, that's
and I, I think perhaps the most important thing is that, I mean, take a research scholar and let's say he or she is working in a lab. They should ask themselves, have I done anything which my supervisor has maybe not asked me to do? Have I thought of something on my own? Some new way of doing something? They should ask themselves this question every day. या मुझसे जो कहा गया मैं वही कर रहा हूँ. So that is important.
so that freedom of thought should be there and
Freedom of thought should be there. If you think of something new, your way to do it, you have to defend it. Yeah, because everything requires resources and money. You can't just be thinking of something which is not going to work.
Yeah.
And not just the research scholars, the supervisors need to have that, foresight to allow different things to happen, to allow.
yeah.
Yeah, it's both ways.
Both
It's both ways, yeah.
So, sir, sitting in IIT Bombay, is one of the most premier institute in India. What IIT Bombay actually fosters in terms of research and then development of research,
I think
which sets it's apart from rest of the
no, IIT Bombay gives you a lot of independence to do what you want to do. And I mean, of course, the infrastructure is fantastic. You can't work in the middle of nowhere. So I think if you are good, that's the only two things you need. Independence, let them do what they are doing with reasonable control, of course, and infrastructure and, when we talk about the infrastructure, we just think about walls and buildings. No, that's not it. Even to like small, we were to buy 100 things every day.
yeah.
To process those orders, we have excellent staff who help us, and there's a mechanism for things which is really nice.
So you can say the overall environment and infrastructure in IIT is at par with the global standards.
Well, the places I have worked, I have not worked in this way in every possible institution in the world, of course, but I don't see, every place is going to be different. You have to, uh, you have to work within certain parameters, and it works for me.
No, that's good. I agree.
It won't be fair to compare IIT Bombay to every other place in the world. There's some good things here, so there's some um, good things there, and there's some bad things here, there's some bad things there. I think it's all
Fairly.
averages out.
Yeah, I totally
Nobody is stopping you here, ऐसा करो, ऐसा मत करो. There's no, I think if you are reasonable, things work.
Yeah, yeah. I think that is more important. You said no, yeah, the basic platform is required, but obviously the thought process has to be evolved. And then I think that is one line I am going to take it from you also. That what one thing I did differently and independently I thought,
Yes,
which the other people are not doing it.
yes, yes.
That sets me different from the crowd.
Yeah. So, I mean, it's not necessary that all the students have to start thinking and they have to mature as early as possible.
So in terms of research scholars, you also seen batches of students have come in the past, and you also see.
Sure.
You seen any evolution has happened in their attitude towards research?
No, I really don't think there's so much heterogeneity in from student to student. I don't think, I mean, that overall the nature of students has changed or they have become better or they become worse. I really don't believe that. I don't think so.
Sure.
Yeah.
Right, sir.
Yeah.
So thank you for all these insights, because this is
Sure.
generally I go to different institutes and it's my inquisitiveness or curiosity, you can say, that I want to dig into what is happening.
This, you're providing us a great platform where if somebody else listens to it and it helps them in some way, or I get some feedback, that is great for me. Yeah.
True. This is actually, sir, for all the like scientific enthusiasts.
Sure.
I'm trying to share whatever I am experiencing and bring it to larger audience. If at least they can take some portion of it and their life changes because of this conversation, I think the success of my platform is there only.
Yeah, that's great. Then, you will be proud of it. I will be proud of it. Yeah.
Thank you. Thank you. With that note, thank you, sir, for
Sure.
your time.
Very welcome and all the best. Yeah.
Thank you. Thank you.